Good afternoon, everybody. We're going to get started here now. So please do enjoy your lunch while I'm giving this lecture. Before I start, I really would like to thank everybody for attending. I've attended many of this myself, so it's kind of surreal to be on the other side of the podium and be presenting to you. So I really hope you enjoy my presentation. And again, thank you so much for coming. This is going to be Serendipity Science, a presentation about our findings in the Fort Worth Botanic Garden. A brief introduction. My name is Alicia Tolley. I was born and raised here in Fort Worth, Texas. I currently attend the University of Texas A&M Corpus Christi, where I am majoring in environmental science, and I'll be graduating this December. Looking forward to it, very excited. And here I am interning at the Britt, mentoring under Dr. Harold Keller and working alongside with Edward Forrester. Just to give you a little bit of a brief methodology, what we did for our project. So what Dr. Keller and I decided to do is that we would utilize moist chamber batch cultures to be able to observe bark samples from various tree species in the Fort Worth Botanic Garden. And what we were looking for is different myxomycetes and fungi. So how did we start? Dr. Keller and I sat down in his office and we had a list of about 14 different tree species, and we decided to pick 6 of them. How did we pick these 6 species? Well, we went back and forth. He picked 3 and I picked 3. I purely picked off of interest. I wanted to look at the Kentucky coffee bean because half of my family is from Louisville, Kentucky. The Osage orange, that weird little ball of fruit you see, I don't know, I always saw it as a kid. So I was like, you know what, maybe it'd be interesting to see what this actually is. And of course, the Texas pecan tree, because why wouldn't we pick our state tree? And Dr. Keller picked Eastern red cedar, bald cypress, and ginkgo biloba because he believed that this is where we would probably find the most myxomycetes, fungi, or any species that we were looking for. So our first species that we picked, like I mentioned, the Kentucky coffee bean. This is one of the dioecious species that we have picked for our sampling. On the left is a picture of the tree, and on the right is a picture of the fruiting body or the bean that you would see on the Kentucky coffee bean. The next is going to be the Eastern red cedar. This is also another dioecious tree, meaning that there is a female and a male tree. And so the female tree is going to have the fruiting of those berries that is actually used to flavor gin whiskey. The weird fruit, the Osage orange tree, another dioecious tree. This is one of the tree species that we also chose. And there is the fruit. It is highly poisonous. You may have heard it be termed as a horse apple. Do not feed this to any animal, cattle, or anything like that. They will die. Very highly poisonous, but the female does fruit those fruits. In the bald cypress, I actually took this photo here in the garden. Now, if you take a close look at the picture, you can see that the trunk of the tree is fairly thin, but once you get to the base of the tree, it flares out. This is because of its buttress root system. Now, that is not to be mistaken by the cypress knees that you see on the outside growing above the ground around the tree. Those act as aerators to the tree because this tree is often found by bodies of water. If any of you have heard of Caddo Lake in Texas, there is plenty of these trees in the water around the water. The next tree you picked was the pecan tree. I also took this photo. This is the tree that we sampled from in the garden. Beautiful, big and tall tree. And finally, the maidenhair, or many of you may know it as ginkgo biloba. The trees on the left are what the ginkgo tree will look like in the fall. Beautifully uniformed golden yellow. We actually have one in our backyard, and I included this picture because my mother loves it when it turns this color in the fall. It is absolutely beautiful. And if you look at the picture on the right, you can see that the venation of the leaf kind of gives it It almost looks like a butterfly wing, but it is two lobes, which is why it's called biloba. Okay, now that I talked to you about the tree species that we sampled from, I want to get into the collecting methodology. We had to follow some very specific guidelines when it came to extracting the bark samples from the tree. The first guideline is that the tree must be 100% healthy, meaning that there should be no dead limbs on the tree and the tree should not be severely cut. Secondly, the tree must be at least 50 feet tall and 3 feet in diameter. The reason why you want those measurements specifically is because you will know that the tree has been there for a while, meaning that it is older and mature, which means it's more likely to have the myxomycetes or the fungi that we are looking for. Oh, sorry about that. Okay, no problem. The third point is that you want to collect a minimum from 3 feet above ground level, or as some may know it as breast level. That is just to be able to ensure that you are collecting not too close to the ground in the middle of the tree where you are more than likely to get proper specimen samples. And then finally, which I believe to be one of the most important guidelines to follow, is that you must ensure that you are not cutting too deep into the tree to harm the tree. And I'm going to talk a little bit more about that. So, what's wrong with these trees? Why can we not sample from trees that look like this? Well, the tree on the left, as you can see, it is missing a lot of foliage, which lets you know that the tree is partially dead. You do not want to sample from a dead tree. The tree on the right obviously has been torn in half. This could be due to animals, a windstorm, lightning, we're not certain. But you do know that this tree will eventually die because it is so severely cut in half, and you do not want to sample from a tree that is in that state. Now, this goes on to talk a little bit more about the point that I believe is the most important, not cutting too deep into the tree. So this is going to be an aerial view of a tree. If you slice the trunk in half, the outside, the outer bark, which is what you want to sample from, is as deep as you want to cut into the tree. Right underneath the outer bark is what they call the living layer of the tree bark, which is the cambium layer. You do not want to cut into this cambium layer because that is when you have harmed the tree and you could cause it disease or it to die. Here is an example of what a harmless extraction looks like versus a harmful extraction. On the left is where we actually collected samples from the pecan tree in the Fort Worth Botanic Garden. As you can see, you do not see the cambium layer or the living layer of tree bark. You do not see any white. You just see where I have removed the outer bark layer. On the right, these are pictures that show the example of reaching that cambium layer or deeper, which is harmful to the tree. Now, after you've collected all of your specimen, your bark samples, it is now time to make your moist chamber cultures. And so the first step that you're going to want to do is you're going to want to collect all of the tools that you need. You're going to need saran wrap, an aluminum pie tin. You're going to need 2 paper towels, distilled water, and a Sharpie and your bark samples. After you've collected all of the supplies that you need, you're going to start to assemble your moist chamber culture. And so how you do this is you take your 2 paper towels, you lay them flat into the bottom of the pie tin. Then following that, you're going to take your bark samples. Some of them you may need to cut, break apart to fit into the pie tin. Some pieces may just fit automatically. In this step, it is very important to ensure that no bark samples are overlapping. They should be all laying flat on the bottom of the surface of the pie tin. This is important because you want to make sure that all of your bark samples absorb that water and are consistently moist while they're in the moist chamber culture. Once the bark samples are laid flat into the pie tin, it is time to pour in your water. Now this, you have to be very careful when you're assembling this part because you do not want to pour the water directly on top of the bark samples. you could wash off or destroy any of the microscopic myxomycete or fungi that you cannot see to the eye. And finally, you're going to cover your pie tin with the bark samples in the amount of water you believe is significant enough for your bark samples with saran wrap. Cover it tightly, make sure the saran wrap is not sagging, and then you're going to write the collection data on top of the saran wrap. The collection data consists of the type of tree that you selected your bark samples from. So in this picture, it may be a little hard to see, but this is the Eastern Red Cedar moist chamber culture we assembled. And so at the top, I wrote the Eastern Red Cedar, where I got the Eastern Red Cedar sample from, which would be FWBG, Fort Worth Botanic Garden. And in these, with this project, we decided to pick 2 different trees of each species. So we had 6 species, but we had a total of 12 trees that we were working with. And so we designated which pie tin was for which tree. So this is for pie tin number 2. After that, you're going to want to write the wet date. And I decided to also write the wet time because you can start to scan your pie tins in as soon as 24 hours. Myxomycetes can develop within that time frame. So now to the good part. What did we find? Let me tell you. Let me tell you. We're going to figure out why it's called serendipity science in a second. So here are highlighted findings on our trees. On the ginkgo biloba, we found a beautiful white grayish stalked Arceria cinerea. On our Kentucky coffee bean, we found what Dr. Keller believes is an undescribed species, a Lycia. It is beautifully sparkly, coppery. This one has a smooth surface, and it can be about 100 micrometers big. And on the Osage orange, we found the Chamatrica cornerea. This is a stalked myxomycete with brown spores. I did not even know what a myxomycete was supposed to look like, what it even meant to look for a myxomycete, but the fact that they can come in different shapes, stalked or sessile, which is what the Lycia species is, meaning that it does not have a stalk. Very interesting, all very different and very unique. Now, let's go to our pecan tree findings. This tree was very successful in terms of having the most species and myxomycetes and fungi that we found. So again, on the pecan tree, just as we found on the Osage orange, we found the Chamatrica species. In the middle, you will see a Chamatrica slide that I prepared. Very, very time consuming, but all worth it. This slide is showing the network of the Chamatrica once I have laid it in the slide and kind of got rid of the spores that were glutenated onto the Chamatrica. So you're going to have the stalk, and then you're going to have the columella, and then the branch work that you see is going to be the callidium. And the little circles that you see are some of the spores that were still remaining, some of them still glutenated, some of them around the actual species. And then finally, we have our serendipitous species, all the reason why it is serendipity science. Okay, so here we have a yellow, hairy, cup-shaped fungus, right? And originally, there was a big question about what this could be, and there still is, but let me tell you what we figured out so far. So originally, we got in contact with one of Dr. Harold's good friend from Harvard University, Dr. Professor Donald Pfister. And we kind of wanted to see if he could tell us what he thought it was. He is a really good general mycologist, and so he originally believed that this belonged to the Cephella group. And going off of that, based off of that, we did a little bit more work with the microscope, trying to extract the spores from this specimen. And we realized that it is a Basidiomycete spores. And so with that being said, we decided to get in contact with another professor that works at the University of California in San Francisco, Dr. Brian Perry. He is an expert over this group. And so we sent him 2 samples, bark that was covered in this species, one that was in moist chamber culture and one that was fresh off of the tree. And so what we did is we decided to send that to him so he could do the DNA and molecular work on the species so we could kind of, figure out where to place this species. And so right now, where we're at in this is either a specimen that has been described 175 years ago and has now been rediscovered, or it is a new species. In his words specifically, he says that this does not fit into any existing genus morphologically. It is not Cephella nor any of the other species that he thought it would be. Right now, he's trying to do further DNA sequencing to see if he can find a family that it belongs to phylogenetically, because as of right now, it appears to be an undescribed taxon. Something interesting about this specimen is that it has all of these filaments, these hairs. And so going back to these pictures, it kind of looks like there's crystals in the middle. Those are the spores. And so all of this is based off of speculation, but it is to be speculated that the filaments are there to protect the spores from any predation or for the well-being of the spores. And so previously I showed you pictures of the species in a wet state, in a moist state. This is the species in a dried state. This species can range from about 100 micrometers to 500 micrometers. In the dried state, it is smaller. In the wet state, it will expand and you'll be able to see the middle a lot better. I just feel that it is so interesting that it's like a little hairy cup fungus. It's just cute. It's kind of cute to me. So these are some of the best pictures that we have. Thank you to Ed Forrester and lots of work under the dissecting microscope. As you can see, this is probably the species in its most perfect form. This is in the moist chamber culture. As you can see, the reflection from the bark behind it. The green behind it is a lichen. It is not a part of the actual species itself. So you can see the hairs a little better on this one. You can see on the right, it is a little more closed. And on the right, you see that little fuzzball looking thing, a very small one. That is a immature version of the species. So it does start off like a little hairball and then it kind of like matures into this little cup with more filaments. And so right now, like I said, we are just waiting to get more DNA and molecular work done so we can see where or what this species actually is. We're deciding if it is a rediscovered species or if it is a new species. And I'm like waiting hands and toes on the edge of my seat, like, oh my goodness, what is it going to be? So very exciting find. And this is exactly why we call it Serendipity Science. We had no idea that we were going to find this. Dr. Keller told me multiple times as I was making the moist chamber cultures that, Alicia, I can't tell you what we're going to find. I don't even know if we're going to find anything, but I know we'll find something. And boy, was he right. Did we find something? So for sure, I'm just almost at a loss for words, even though I'm giving this presentation. That will be the end of my presentation. I would love to thank everybody that has helped me so much. Of course, Dr. Harold Keller, Edward, Bob, Aaron for helping me set all this up and getting me through the process, Morgan for helping us get the water. We couldn't have done this without you, and Dr. Brian Perry, Dr. Donald Pfister from Harvard for giving us our first insight of what this could potentially be, Ashley Bales for helping us, and they're actually the Dr. Keller and Ashley originally made a video and a paper about moist chamber cultures. And I would like to thank them and her for giving me the idea and letting me take on their project and their idea. Seth, we would not have gotten any tree bark samples without you. So thank you so much. That go-kart came in handy because walking in the Botanic Garden when it's like 100 degrees is not it. And then of course the Brit, because I would have never had an opportunity like this if it wasn't for the Brit and if it wasn't for Dr. Keller being there. So thank you so much. I'd also like to give a bigger special thank you to Dr. Keller. I'm gonna get emotional, hold on. Thank you so much for this opportunity. This is just, he's probably gonna kill me for putting these pictures up here, but this is when we were collecting the tree bark samples. As you can see, I had no idea he was gonna be sneaking and give me these little bunny ears. And then the middle picture is me, like Seth was like, okay, Dr. Keller, let's be serious. I said, what is he talking about? I'm serious this whole time. And then he shows me the picture and I'm like, Dr. Keller, you were giving me bunny ears. He's like, yeah, I was. And then of course that's our happy serious picture. That is actually the pecan tree where we found this amazing fungus from. And so we were just collecting and looking at everything. You could actually see it with the naked eye on the tree bark. We did not know that at first. We were just taking tree bark samples the first go around, but when you go back, you can vividly see the yellow on the tree bark. And so that is our gift from above, that pecan tree. But thank you so much, Dr. Keller. This opportunity was absolutely amazing. I cannot wait to continue to write, work on a publication with you, and figure out what this thing is. Thank y'all so much. Erin is going to be passing around a microphone to whoever has any questions, so please feel free to ask as much as you want.
Awesome talk and very cool discovery. Thank
Thank you.
That was amazing. I just had a quick question about, in particular, whether or not when you do the moist chamber culture, do you see changes over time in terms of the, like, I guess, density of the communities that are on the bark that are, growing the slime molds? And do you see differences like between the communities on the outside of the bark versus on the inside? I've seen the moist chamber cultures in Harold's office, but silly enough, I haven't actually asked to take a closer look, which
Right.
seems crazy. So
When you say on the other side of the bark, you mean like underneath, if you flip it over, if you see something underneath?
yes,
Okay,
exactly.
for sure.
So whether the outer or the inner have
Okay.
differences.
Yes. So one thing about the tree bark is that you can re-wet the tree bark over time because it will dry up eventually. The water does not stay forever, especially opening and closing, taking the saran wrap on and off. And so one of the things that you have to watch for when you're re-wetting and opening and closing, there's a lot of contamination that can happen. There's also you have to be cautious of not re-wetting too much or directly on top of the bark because that can affect your growth rates and what you see and what you don't see. And so I think once we re-wet, when we re-wet, that's when we saw a lot, specifically the fungus. No, that had been there from the beginning. The only thing we had to worry about with that is the green mold because that's a contaminant. You will have to throw your whole moist chamber culture away. But other than that, it's pretty uniform. Yeah, of course. Good question.
Just wanted to ask, where can we get updates about what you find about this new Basidiomycete?
Right. Good question. That's a good question. So I definitely think I will, that might be a question for Dr. Keller, but I'm going to assume that once we get more information and actually are able to create a publication, it will be connected to the Brit website.
Very cool.
Yes.
And are you planning on working more with fungi or mycelium in the future?
That is such another good question. So originally what I wanted to do with my degree, with my Eastside degree, it is a concentration in marine and coastal conservation. So I wanted to work in the water with stingrays and be swimming in the Little Mermaid. But, somebody asked me the other day, they were like, do you think you'd be able to identify this and just leave this world alone, leave the fungi, the Myxomycete world alone? I think the answer is no. I might be an accidental botanist just like you, Dr. Keller. We're gonna have to see. Thank you. More serendipity.
Great talk and great project. Congratulations, and thanks for taking the time to share it with us. I'm
Thank you.
very proud and I'm very happy to see
Oh, thank you.
and to meet you because I wasn't here for the summer. So very nice. I have two questions.
Okay.
The first is, what is a myxomycete?
Okay.
How you probably say it, but if you can. And the second one is, if this specimen that you have is a new rediscovery, can you tell us about, like, where was it collected before, like 150 years ago, or is there, like, what's the story behind the other specimen?
Of course,
if you
of
know it.
course. Yes, I do actually. And so Ryan Perry is actually the person, the authority on this group, like I said. And so he has done his digging and he was able to find where he believed that this species may have been discovered 175 years ago. And so let me pull up that information for you so I can read it exactly verbatim for you, because it's kind of interesting to wrap your head around the fact that a species can be rediscovered, especially from over 100 years ago, almost 200 years ago. So let me get that information for you right now. Okay, so he says that this species is known from a single collection made from Curtis Woods in the late 1880s, someplace in Texas. All the descriptions of this taxon are much too brief to determine if it is microscopically similar to your collections. Sorry, I thought it went out. So from also what we understand is that the person that went and made these collections went on foot from El Paso to San Antonio, I believe. Right, back then, I don't know how they would have been able to do that. I cannot imagine. But what we are speculating is that maybe they have found this species from living bark, maybe that was already on the floor in the process of dying. We're not too certain. There is not a lot of information on where this species was maybe first described. That's what makes it a little more interesting, a little more serendipitous. Yes. Mhm.
What is a myxomycete?
What is a myxomycete? I will let Dr. Keller answer that in detail for you.
I was afraid somebody was going to ask that question. A myxomycete is a microorganism that has a life cycle where it forms fruiting bodies, which you have seen, that have spores that germinate to give myxomebian swarm cells and eventually form a plasmodium that creeps across the bark or the leaf litter and eventually forms the fruiting body. That briefly is the life cycle. So it's never really entirely animal. It's never entirely plant. It's a new form of life. And so it's very different. And it intrigued me when I was at the University of Kansas a long, long time ago. I collected my first myxomycetes, and I've been working for more than 55 years on this group. I have a question for you. I'm going to keep this.
Yes, sir.
All right.
Uh-oh.
Now, everybody that goes to these talks knows that I ask a question.
Yes.
So I'm going to ask you a question.
Okay.
What is the most interesting thing you've learned during your mentorship here at Brent?
Wow, a lot of information. How do I pick one? Let me think. Well,
Be careful.
yeah, I know. Oh my gosh. Well, definitely, I think I've learned overall that a job or research like this is never just science and never just people. It is both, inevitably. And to me, I've had the most beautiful experience when it comes to both. And so partially, one of the most interesting things I've learned about was you and everything you've done. I cannot believe that you know, somebody has literally dedicated their life work to a group that maybe arguably could not be that much of a high in the hierarchy when it comes to what is studied in the science field, but it's so interesting. How could it not be? And on the scientific side, I had no idea how to really use a microscope. Thank you to Ed Forrester. Did not know what image stacking was. I've only ever seen the pretty images in my science textbooks that are this thick in school, you know. I didn't know how to do that. And I didn't know what it actually took to actually be able to do research and commit to research. A lot of time, a lot of hours, a lot of trial and error. And so I think overall I learned how to be a little bit more of a scientist while I was here. And I think that's the most interesting part.
Another question.
Yes.
When you graduate, what do you plan to do?
Well, when I graduate in December, oh, Mother thinks that's a great question. I bet she does. Well,
That was her mom.
well, I will be applying to grad school in November at the same university. I plan to go into the same degree, environmental science. There is a thesis route you can take or a project route. I plan on taking the project route. Of course, Alicia will have to start looking for her big girl job now. Right now, I work in as a secretary in the dean's office at school for my college, and so there's a lot of what ifs right now, but I know I'm ready to get there. Do you have another one? You have to have another one.
Well, this is a work in progress. I've repeated that many times, and that we still have a lot to do. Brian Perry is in Alaska. He just had COVID-19, and he's in Alaska and tied up, but we're waiting for the molecular data to tell us what we've got. We don't know what we have. You'll notice the photograph showed it was cup shaped. That looks like an ascomycete, and that's what I thought we had. But it's a basidiomycete. That's the mushroom group. It's a totally different group. It doesn't look like a mushroom should look.
At all.
And you see the pictures there. Those are Ed Forrester's photographs. And we have beautiful, beautiful photographs. Ed calls it the hairy thing. I think that's a good way to refer to it.
I think we have two questions.
Okay, I'm sorry.
No, it's okay, doctor.
Yes, for the stream.
Oh, we have to. Oh, yes.
Something. Thanks. Great talk. Great.
Thank you. Thank you.
Interesting findings, Alicia. And I'm wondering, do you have SEMs of the spores?
We do not have SEMs of the spores, but we do have microscopic pictures of the spores.
Oh,
Yes.
like microscopy?
Did you get some pictures like that of the spores, Mr. Ed?
Working on it. Okay.
Yeah.
Because doesn't that tell you that it's a basidiomycete
Right.
rather than
Exactly.
actomycete?
That's exactly how we figured out that it was a basidiomycete from the spores. Yes. I believe you had a question.
Thank you. That was a wonderful talk. So
Thank
great.
you.
And the pictures are wonderful. All the work
Great,
is immense. Question and an offer. The question first is, in each of the bark samples you had in that single moist chamber, did you find the species on all of them?
So pertaining to this,
Mm-hmm.
a majority of them, yes, I would say 75% in each chamber that did have it. Yes.
And in both pecan trees or just one of the
Just pecan tree number 2. Yeah, number
Okay.
1. Actually, I have a picture of both. Pecan tree this is going to be pecan tree number 1. Next to the eastern red cedar. I do not recall where exactly it is in the where is this? Do you remember, Seth, where this exactly is? In the floral garden? Yes. So that one did not, that tree was actually unsuccessful. Like we didn't find any myxomycetes nor fungi, but on pecan tree number 2, that's where we found our specimen. Oh yes, I think I mentioned earlier that you can actually see the fungus with the naked eye on the tree. So yeah. I did.
So the second is an offer, and because you've got these vouchers of this Basidiomycete, it would be interesting to pair it with a voucher of the tree itself. It's part of our project to
Okay.
sample our living collection anyway. So I would offer that if you have time before you go back to school or when you're on break at some point,
Uh-huh.
we can go voucher those collections,
Okay.
or I can do it for you, but then we can cite that your Basidiomycete specimens came from that tree
Okay,
that we
that-
have in the herbarium.
That
So,
would be wonderful. Yes, of course. We will have to figure that out for sure.
well done. Thank you.
Thank you. I believe my mother had a question. Uh-oh.
It's just for us layman people that don't know half the vocabulary spoken today.
Okay.
But does it harm the tree? Do these things harm the tree?
No, it does not seem to harm the tree. It seems to work symbiotically on its own. Yes, that was a good question. Okay. She's like, run, run, run across the room.
Congratulations. The photos are very beautiful, but I tell you that if you look at the lichens under the microscope and take photos, they will also be beautiful.
Oh, I see.
First,
Understood. Respect.
My question really is regarding the collection methodology,
Okay, of course.
because you mentioned a while ago that you concentrated on healthy trees, right? Did you delimit your collection only to the fungi, or did you not attempt to look for slime molds? Because I had a look at slime molds also in the Philippines. In the Philippines, we have a lot of slime molds, and every time we go to the forest, we do see a lot of slime molds, but we usually see them in rotting trees, logs. So we always choose to look at the rotting logs
see.
because you will really see a lot of
Mhm.
slime molds there. That's why I was really wondering why you really just delimit your collection to the healthy barks of the trees.
Well, I know for a fact that we want to sample from those trees just in case. What if we have to go back and get more bark samples? The tree needs to still be there and it still needs to be healthy and intact and a good environment to sustain the myxomycete or the fungi that we are trying to look at. So I can understand that that's where slime molds flourish. This is where myxomycetes and fungi would flourish on a healthy living tree. Yeah.
The myxomycetes that occur on the bark surface of living trees, I coined the term corticulous myxomycetes. They occur there and nowhere else. So there's some kind of interaction between the bark of a living tree and these species that enable them to survive, grow, and develop there that they can't develop on rotting logs or rotting leaves where you typically find myxomycetes. And so that's why we sample. And by the way, she didn't mention that her samples from these trees, except for eastern red cedar, are the first time these trees have ever been sampled from our Fort Worth Botanic Garden. So they're really special. And she called it Serendipity Science because we didn't expect to find this. And we still don't know what it is. So it's a work in progress. Sorry.
Oh, 2 questions from the chat. Exciting.
Did you answer the question? What's the most interesting thing you've found since you've been at Britt?
What is the most interesting thing I found? How could it not be our fungus? Look at her. She's so it's beautiful. When you think of fungus, like you're like, ew, like black mold or, no disrespect. But that's at least what I thought until I was educated on this group or myxomycetes in general. So I don't know, it reminds me of the sun. It's so pretty. A little tulip. I feel like that was the most interesting. What were the questions from the chat?
So from Natch, we have, if this is a new species, what would you consider as the name? Yourself?
Okay, because I've been thinking about, can I name this Alicia Tolley or something? No, I'm just kidding. I'm just kidding. No, definitely. I'm certain it would have to be, since We haven't necessarily talked about this, of course, because we don't know about what it is. But I'm thinking in my head, if it's found in Texas, and the only other trace of it was in Texas, I would probably put Texas in there somehow. I would love to name it Alicia. That'd be kind of cool, or Celine, or something like that. But that's a work in progress.
And then our second question comes from Andy. How long, actually, a great presentation. How long did the research take, and how do you gather these beautiful images of the fungi?
That was a great question, Andy. So really, we did have a short amount of time to conduct this project. We could have kept going and probably found so many more things because, like I said, when you re-wet the bark, things appear that weren't there at first or won't be there for another month. That was the case for the Lycia species that we found. Not for the fungus. The fungus was there from the beginning. But it took the entirety of my internship to conduct and collect all this information. But under my impression, I'm certain this could have gone on for a year, for as long as I'm concerned, because you just keep finding more things. Of course. Oh, how did we take the photos? Okay, yes. So two ways we took the photos. Under a dissecting microscope and under the image stacking program that we used. Ed Forrester was a big help with this, taught me how to image stack, taught me how to do the image stacking and the dissecting microscopic photos. And so really good technology and really good luck is really how we got the photos.
Is the fungus not directly growing on the lichen? Not
Okay.
on the bark?
No, so
It's a lichen.
it's
What I'm
okay, it's
trying to
okay.
say, it could be a lichenicolous fungus, you know.
That's a good question. Well, so in this picture, you can see that the fungus does not only grow on top of the lichen, it does also grow on the bark. And so the question is also, what is attaching this specimen to the bark? Speculation says that there are a lot of small rhizoids that are attaching this specimen to the bark, but we do know that it is not stalked, so it is sessile. Any other questions? They were all really good questions. I appreciate them. Oh, thank you. Thank you so much. Thank you so much for coming out, everybody. I really appreciate it. I've had a wonderful first time experience presenting and doing something like this. And I can tell that it took your interest, and I really appreciate that because that was one of my biggest goals. I wanted to be able to educate properly and be able to provide information in a presentation that would capture the audience and have you thinking a little bit more, or make you think maybe my kid could do a moist chamber culture. What if we find something or anything like that? So thank you so much to everybody who came. I really appreciate it. And I really look forward to continue to working with the Brit and Dr. Keller. Thank you.